Hello everyone. Today we are going to learn about acid-fast stain or Ziehl-Neelsen stain. So acid-fast stain was first developed by Franz Ziehl and Frederick Nielsen, but was first employed by Paul Ehrlich in 1882. So this is Franz Ziehl who first employed carbolic acid as a model. This is Frederick Carl Adolf Nielsen who replaced the primary stain with basic fuchsine. Though Franz Ziehl and Frederick Carl Adolf Nielsen developed the first primary stain, the complete preparation was first applied by Paul Ehrlich on Mycobacteria in 1882. Where? In Germany. So coming to the acid-fast staining, so here it is a differential staining procedure that divides the bacteria into two groups. Now before that, it is to be noted that it is a widely used staining method that is used for Mycobacteria. In the early 1890s, it was referred to as Ziehl-Neelsen acid-fast stain only. But now these days, it's commonly known as acid-fast staining. So basically, it divides the bacteria into two groups. One is acid-fast stain and the other is non-acid-fast stain. Now, what are the reagents that are required in this procedure? So, the reagents that we need are primary stain, decolorizing agent and a counter stain. So, primary stain here, carbylfuchsin is used. For decolorizing agent, acid alcohol is used, which is composed of 95% ethanol and 3% ECl, and a counter stain, methylene blue, is used. So this is the carbylfuchsin and this is methylene blue. Now coming to the procedure of the acid-fast staining, we have in step 1, first the bacterial smear is prepared on the slide and it is air dried, followed by the addition of the carbylfuchsin on the smear. Now this carbylfuchsin is kept for 1 minute and then it is heated for 3 to 5 minutes more so that it starts to steam. So when the evaporation starts, more amount of carbylfuchsin is added. So this heating, what it does, it enhances the penetration of the primary stain into the waxy cell wall of the bacteria. Since the Mycobacteria have a different cell wall composition than that of gram-positive and gram-negative bacteria, so that's why this acid-fast staining method is used, right? And that's why the heating is necessary because they have a much more amount of waxy substance or the lipid material present in their cell wall. So that's why heating is required. Then it is washed with distilled water and decolorized with acid alcohol for 1 minute. Again it is washed with distilled water. After that, methylene blue is applied for 1 minute and after that it is washed with distilled water. Then the smear on the slide is air dried and observed under microscope. Then coming to the pictorial representation of the acid-fast staining, which will make your understanding more clearer. So in the step 1 we had the bacteria, which we have made the smear on the slide. It is then air dried and then the carbylfuchsin is added onto it. And after the addition of carbylfuchsin, it is heated on the flame for 3 to 4 minutes just to start evaporation to ensure that the carbylfuchsin is entering into the waxy cell wall of the Mycobacteria, right? So that's why the heating is done. So next it is then washed with distilled water. After that it is given acid alcohol for 1 minute just to remove the stain that are not required, that are not taken up by the cells. Again it is washed with distilled water. After that, methylene blue is added for 1 minute and after 1 minute, it is then washed with distilled water again. After that, the slide is again air dried and then it is observed under microscope. So this is the whole pictorial procedure of the acid-fast staining. Now coming to the observation under microscope, what we'll see under the microscope is that the acid-fast stain, they will retain the carbylfuchsin and they will appear red in color, somewhat like this. And the non-acid-fast bacteria will retain the methylene blue and appear blue in color, somewhat like this. Now what is the reason behind this acid fastness of the bacteria? So coming to the mechanism of the acid-fast stain, so it depends basically on the composition of the cell wall. Like I said previously, that the cell wall composition of the mycobacteria is very much different than that of gram-positive and gram-negative bacteria. So some bacterial cells, for example, these mycobacterial cells, they are surrounded by a covering on their cell wall. Outside the cell wall, they are surrounded by an extra covering which is composed of fatty acid or waxy substance, which makes it non-permeable. Right. So we'll see the picture here. So here, the outer membrane we have just like that of gram-negative bacteria. The outer membrane itself is composed of lipids. Then we have mycolic acid, the thick layer of mycolic acid we can see here. And after that, a thick layer of arabinogalactan is present. After that, there is a peptidoglycan. Then after that, there is a cell membrane. So we can see there is a huge layer of lipid. Huge 1, 2, 3, 4 layer of lipids are there. Okay. So it's a very thick layers of lipid present. So that is why the normal gram staining procedure is not applicable to identify acid fast or non-acid fast bacteria. So that is why the acid fast staining is required. So this mycobacterial cell wall, they are rich in lipids due to the presence of mycolic acids. Now these organisms, they are not readily stained, but once the stain is introduced in the cell wall, it becomes very difficult to decoloриze it even with drastic decolorizing agent. Even the decolorizing agent like acid alcohol, they cannot even remove the color once they are stained. Okay, so that is why they are called acid fast stain. Why? Because they are resistant to decolorization, right? So what are the examples of this group? There is mycobacterial species and the other is Nocardia. So in this video, we have learned about acid fast stain, scientists who first prepared it, the reagents used in this procedure, the main procedure, and the mechanism behind their acid fastness.